We're going to continue our genetic story. We're going to move on to understanding in more depth how to go from a genetic blueprint on DNA to actually making a protein that can have an impact on the traits of a cell. So this is going to be another section of Chapter 8 that we're going to have to chop into multiple video clips so that we don't have single video clips that go on and on and on and on and drive you crazy. Turn on my iPad here so I can do some annotations when needed. All right, so flow of genetic information in cells. So as I mentioned, we've already talked quite a bit about DNA, and we've talked about how DNA molecules have locations on them that are called genes. And many genes ultimately function as blueprints or codes that will tell the cell how to make a protein of some type. And cells are going to have thousands of different kinds of proteins that they need to manufacture to carry out all of the work that they need to do. Many of them are enzymes. You've learned about membrane proteins before that transport things, from inside the cell to outside. And you have proteins that play many other roles in cells as well. All right, so think about these guys as being necessary to ultimately get the structures and the functions of a cell and give the cell or the organism its traits. All right, so we have an intermediate in between DNA and protein down here. There are intermediate molecules that are called RNA. All right, and so there's more than one type of RNA in a cell, but RNA molecules are copies of a DNA strand. So RNA is a nucleic acid like DNA, but remember you have those little differences that we talked about between DNA and RNA nucleotides that are used to build them. And when you make a RNA copy of a stretch of DNA, you make a complementary copy of some stretch, that process is called transcription. So you're copying the DNA or transcribing it into an RNA molecule. And the main ones that we'll talk about are right here. These are called mRNA or messenger RNAs. Messenger RNAs are copies of genes that code for proteins. So messenger RNAs go to these structures. These are ribosomes. Remember we've talked about before how ribosomes are the machinery that build proteins in cells. And then a ribosome will read this messenger RNA and link amino acids together to build a protein. And there's another type of RNA that we'll talk about as well called tRNA or transfer RNA. And you'll notice down here that they're present in the ribosomes as well. They're going to play a role in the manufacturing of the protein as well, as we'll see. And there's another type of RNA over here called rRNA, which stands for ribosomal RNA. And ribosomal RNA molecules are present in this blue structure here is the ribosome, the protein-making machine. And ribosomal RNAs are really components of the ribosome. They're building blocks of the ribosome itself. So they're going to be in here in this blue gamish. But so all three types of RNA there are involved in making proteins. And in order to make these, you've got to have genes on your DNA that get copied or transcribed to make them. So RNA molecules are going to come from DNA through a process called transcription. Now, more recently, the last 10, 15, 20 years ago, we have come to appreciate that there are other types of RNA molecules in cells. It used to be that these were the three that you learned about and that was it. But there are some others called regulatory RNAs, and there are different types: microRNA, interfering RNA, antisense, riboswitches. And we're not going to get into those types of RNA in this class. I think that's getting outside the scope of the class. But I do want you to be aware that they exist, and you've heard the terminology, and you'll read a little bit more about those in your textbook as you're going through the reading assignment. But they're regulatory RNAs because cells very carefully regulate what proteins they make. It takes a lot of energy to produce those proteins. Let's say this protein is an enzyme that's needed to help you catabolize a type of sugar, to break that down. Well, if that sugar's not around in your environment, you don't need the enzyme. So you don't want to go through these complex processes in order to make that particular enzyme. So in some instances, regulatory RNAs get involved in whether or not you're actually reading a gene and using it as a source of information for producing a protein. That's what genetic regulation is all about. And we will talk about that a little bit more as we get into the chapter. Okay, so now for the most part, as we go through genetics, we're going to be thinking about how you have a gene on your DNA, and that's going to get copied into a messenger RNA molecule. That's called transcription. And then that's going to get read on a ribosome, this blue structure right here, and that's going to lead to the production of a protein. So going from the messenger RNA to the protein is a process called translation. Unfortunately, transcription and translation sound a lot alike, and you just have to remember that transcription is going from DNA to RNA, and translation is going from messenger RNA or mRNA here to protein. You're changing languages. That's where the term translation comes from. DNA and RNA are built from nucleotides, so that's nucleotide language. Proteins are built from amino acids, completely different types of organic molecules. And so that protein language of amino acids, we're switching as we go from messenger RNA to protein. So that process is called translation. We're changing chemical languages. Here's another look at this. This is sometimes called the central dogma of genetics. It's kind of an older phrase that has been used. And what that tells us, just what we've talked about before, genes on DNA undergo transcription, and you wind up with a messenger RNA copy. That goes to a ribosome and undergoes a process called translation, which produces a protein. And that protein, in some way, affects the phenotype of an organism, a trait or traits, depending on what that protein's job may be. So if we look at this a little bit more carefully, here you've got a gene on DNA, which is a bunch of A's and T's and G's and C's. You've got a double-stranded molecule, complementary base pairing. When you do transcription, you make a copy of one of those two strands. So if you look really carefully, this messenger RNA copy, a single-stranded molecule, looks just like this strand of DNA, but where you have thymines here, you have uracils here. So you have U's in place of the T's. So that's transcription. We now have a messenger RNA. This goes to a ribosome, and this series of A's and U's and G's and C's gets read on the ribosome, and that tells the ribosome what amino acids are going to be bonded together to build our protein. I know it's saying polypeptide over here, a chain of amino acids. Another name for that is a polypeptide, but protein also means that as well. And a lot of times I just tell students to think of when the polypeptide, the chain folds up into its 3D structure, it becomes a protein. Okay. I've got a link here that you might want to check out. This is another one of those activities on the Learn Genetics website, which goes through the how you go from a gene on DNA to the trait of a firefly glowing. So that's another good activity for you guys to go through for helping you understand going from DNA to RNA to protein to trait. So I encourage you to check that out. As I just mentioned earlier, all right, so proteins determine phenotype, the traits of an organism. How do they do that? Thousands of different kinds of proteins are made in a typical cell. So some play roles in a cell's structure. Maybe you guys remember from Biology 201 learning about actin and myosin proteins in your muscle cells that are responsible for muscle contraction. And they also fill up the bulk of the interior of a skeletal muscle fiber. So in that way, they're playing a big-time role in the composition of the inside of that cell. Enzymes, we've talked many times about those. Those are generally proteins. Now we just talked about how there are RNA molecules that function in whether or not a gene gets read, and that's genetic regulation. There are proteins that do that as well, so they play roles in as genetic switches. Another term for this sometimes is gene expression. I want to go ahead and bring that up because you'll see that phrase used in your reading. If a gene is expressed, it means you're going from here down here to the trait. So you're going through transcription and translation. You're building the protein, and the protein is carrying out its job, so the cell has that particular trait or characteristic. That's called gene expression. So genetic regulation determines whether or not a particular gene is expressed. And again, expression means the trait or the characteristic dictated by that gene can be observed. Many proteins are membrane transport proteins, like sodium channels, or remember the porins that are present in the outer membranes of gram-negative bacteria, and so forth and so on. Motility. If you're a motile bacterial cell, what structure do you need to be able to move around? Hopefully you're thinking a flagellum or flagella. Those are built from proteins, mostly a protein called flagellin. So cells that are capable of moving around, there are proteins responsible for making that happen. So guess what? You have, if you're a motile bacterium, on your DNA, you have a gene for flagellin proteins. You make those proteins, they assemble into the flagella, the flagellum or the flagella that will allow you to move around. If you don't have that gene on your DNA, you're not going to be a motile organism because you don't have the genetic blueprint that's going to tell the ribosomes in that cell that they need to build flagella. All right, so that's kind of just getting us used to this concept of moving from genetic blueprints on DNA to actual proteins. In the next video lecture, we'll start taking more of a detailed look at that transcription process, and then after that, we'll begin to talk more about the translation process.